Okay. So yesterday we talked about some of the general properties of bacteria and how do we categorize them and what are different ways that we can empirically assess infections from gram-positive and gram-negative. And some of the structural elements I just touched upon very briefly, but I will go in detail. As I said, when you come for the lab, we need to know the details in terms of chemistry, biochemistry for the drug action. So those of you who have seen the post on Moodle, it would give you some of the YouTube videos for antibacterial actions and some of the basis of other structural similarities and dissimilarities there. I also noticed that the one lecture posted as the first bacterial classification is a little bit different than the one I presented in the class. So I'll repost that. Okay. So I'll try to repost that. But anyway, I looked at today's lecture. I don't think there's any difference. So you can pretty much use the same version that's there on Moodle. Okay. So what do you see here? How many of you what did you say? Virus. How many of you agree? I mean, if you've gone through the lectures, you pretty much know. So you can see from here a very symmetrical, like what do you call it, eagle landing on the moon type of a thing. And the point I'm trying to make in this particular lecture is this is a virus called bacteriophage. Some of the things I used to wonder, you ask yourself, do bacteria get infected as well? Yes, they do. So viruses have to be smart enough even to infect bacteria, right? And also when infections come, you also want you to keep in mind, by infection would mean that transmittal of genetic material. So that's the point I'm going to discuss today, bacterial genetics, and rest of the stuff is pretty preliminary. I'm pretty sure that you already know that. And there's no kind of rocket science there, but we will go through the physical and chemical growth requirements. What's the effect of oxygen? But I would rather spend most of the time on genetics, bacterial genetics as well. And the whole idea is that if viruses infect bacteria, so that is a transmission in terms of genes. That's what you need to realize, because both of them have genetic material. Okay, so that's something we have to keep in mind. And I'll go through this in detail today. Okay, old slide that you see in basic mitosis slide in your biology as well. Like any cell, bacteria will also divide. So that's like a binary fission. It's going to divide into two, and like any other cell that you know in biology is that it's going to be equally divided. Like we say daughter cell, we say daughter cell. So these are identical daughter cell normally. And then this is a splitting time that you can see from here. All those typical mitosis events will happen where you see a cleavage and then, you know, chromatin going in opposite directions, split, and so on and so forth. But the point is that for every bacteria, there's a time for division, right? And that is the time we say in terms of doubling time. So one bacteria doubles into two, we call that doubling time. It varies, and we'll discuss that in different types of bacteria today. And the most other important thing is that all these events which are happening in division, like any other cells that divide, proliferate, it is pretty much regulated. That's another thing that you have to keep in mind. So the chances are by far that as we say, like daughter, like mom. So mostly the same genes, mostly the same information will be passed. Okay? And things will get confused when we talk about bacterial resistance. But what you need to find out that this whole event, there is a synthesis of protein and there is a RNA there, the genetic material equally divided among the two. Okay. Now this division, as I said, I say daughter cells, but I didn't label any male, female. But as we go along, I'm going to start talking about male and female. I'm going to talk about mating as well. So that's going to be a little bit different as compared to what the initial splitting of cells in terms of biology is concerned. But what you need to know is that this division requires growth factors. Right? So there's a growth factor, and bacteria are selective in that. Some of the bacteria need a specific growth factor. Right? So if you provide them growth factor, they are going to divide. But what you need to keep in mind is, as I said in the red ink over here, once there's a signal for them to divide is initiated, it must run to completion, even if all the nutrients have been removed from the medium. So that's something you have to keep in mind, because like signaling, signal come, divide, and they start dividing that. And also keep in mind, as I said, one of the classes of antibiotics that we use are called bacteriostatic drugs. So this means at the best, we just stop multiplication. Okay? So keep that in mind. Now, the other important thing is that I don't want to go in detail, that if you know the structure of bacteria, with all this bacterial membrane, with all these differences between gram-positive and gram-negative, with all those peptidoglycan layer and so on and so forth, everything is divided into half. There is no kind of handicap or deficiency in terms of division that one gets different information than the other, or one gets partial cell membrane as compared to other things of that nature. Everything is divided. Okay, that's another important basic principle that you have to keep in mind. And as I said earlier, the most important thing that we are interested in is, as I said earlier, the genetic material, the chromosomes. Right? And the chromosomes will divide equally, so all the information for requirements are pretty much the same for bacteria. So what I'm trying to say is that if there are 100 people and each one of them get infected from Staphylococcus aureus, we're talking about the same Staphylococcus aureus by far in terms of the basic characteristics. But the question you have to ask yourself is, when do the change begin, and why do the change begin? Because we want to address something which is coming up in terms of antibiotic resistance. I think people will tell you that the way things are going, all our antibiotics are going to become resistant. So then we'll have nothing to fight infection. So that is the scary part that we need to deal with, and we need to understand the mechanisms. And also, bacteria, when they grow, so we can plot the growth of bacteria in terms of hours, depending upon the doubling time. And we can look at the log growth. So they grow in a log growth. Is a very exponential growth, as we say, very fast. Okay, so let's say, for example, plot over here the bacterial population. So you can see for most of the bacteria, we can divide them into 1, 2, 3, 4 phases naturally. And remember, these are the phases without antibiotics. If I was to take bacteria, which you will do when you come for the lab, you grow them and you can chart them because we can count them. Okay? So the very first thing over here, you say lag phase. This is log of the bacteria, a log phase. What is happening is you don't see any increase in the number because there is an increasing activity which is happening within itself. You know the biology of the cell, something happening within the cell, that things are maturing and duplicating before they split apart. Okay, so you need time for that. We call lag phase, where you don't see one cell divide into two daughter cells. So we go through for that, and that's different for different. So we say lag phase. Then there's a logarithmic or exponential increase. You can see it goes all the way to log phase. And why do we need to know that? Because we are going to use antibiotics to hit that particular phase. Then all the bacteria go through a phase we call stationary phase. They stop, and then they go for a decline. So they also decline in terms of logarithmic state. So this is a basic concept of the growth phase of bacteria. They go for lag, log, stationary, and death phase over time. And that time varies. Okay? Now, there's another thing for the same thing. You can see in the lag phase, exponential phase, where the cell number is increasing. This slide is only difference is just tell you that the cell number is increasing. So remember, we are saying bacteria is a cell as well. Virus is a cell as well. You are a cell as well. So everything comes down to a cell with a membrane and all these organelles which are there. Okay, and there's a stationary phase, and then naturally it should decline. Okay, now the question we have to address is, just imagine for a while, time, I would say Alexander Fleming discovered penicillin 40s probably. So before that, what was happening? There was no antibiotic at all. That's why the biggest discovery was antibiotics. So people knew that bacteria were dividing, and they wanted to find out what are the physical characteristics so they can either control or kill. That was the whole idea. And that's basically the basis of the food microbiology. And that's been there ever since human beings started cooking food, or human being, started looking at this thing. So there are some physical requirements even today that would limit the growth. Temperature, right? So if you boil, bacteria should die. pH, osmotic pressure, pretty much for every cell. So these are still, and you will wonder, how do we use that? Well, we use, even if not use it in infected people, but we use it in food, for example. We cook our food, boil our food, freeze our food. They're all good nature. And why you ask yourself, why do you do that? Because you want to stop spoiling of the food. And how do food get spoiled? Because of bacteria. As simple as that. Okay, we still have that. Okay, and then again, temperature. Again, some of the bacteria want to grow at very specific temperature, so you can kind of go around. Some of the bacteria, you can't even kill them by boiling. Some of the bacteria at the best will stop growth if your food is infected. For example, you put infected food in your freezer. So what best would happen is that they'll just go to the stationary phase. Once you take them out, it will still start growing. Okay, so there is a physical correct for temperature. And you can see from here, there's a rapid growth of bacteria. Most of the time, they also produce toxins as well. As we hit higher and lower extreme, the bacterial behavior in terms of growth may vary. The idea is that at higher growth temperature, it destroys, kills most of the microbes. At lower temperatures, most of the time, it will stop the growth. We just freeze them. Okay, but optimal temperature, as room temperature, as our body temperature, at most of the temperatures we keep our food in. I mean, simple question: if you leave your food on your dining table, it's gonna stay good for a while. If you put it in the fridge, what's the temperature of the fridge? Huh? You're not sure? What's the temperature of the fridge? You use fridge, right? I'm talking to you. What's the temperature of the fridge? You don't know. Interesting, yeah. 2 to 4 degree. 2 to 4 degree. What's the temperature of the freezer in your fridge? You guys are not sure. Very interesting. -10 to -20. Yeah, something like that. And in our labs, we have something like -60. Right? So we kind of manipulate temperatures, but at least you should know there is a half-life for food contamination if you were to freeze it versus put it in the freezer, put it in. So there's a temperature effect, okay? pH, again, is important because most bacteria go for 6.5 to 7.5, and we have like 7.4 pH normal, and more than yeast go between this pH, and then again, some of the bacteria we call acidophils. Acidophils are those bacteria, phil means attraction, they have an attraction for the acids, so acids not going to kill them, so they can sustain acidic environment. So there is an effect for pH. There is an effect of osmotic pressure. We had effects of, we talked about basic biology. I'm going to go in detail. We already discussed that. If you change the salt concentration, it's going to break apart the bacteria, and it still follows the same rule. Bacteria have a growth requirement as we have, CHON, human beings, living cells. So we have to have nitrogen. Bacteria also need nitrogen. They need sulfur, they need phosphorus, because many of them are important part of even DNA or RNA or genetic material. If you don't have phosphorus, then you're in trouble. So proteins need to do that. So that's pretty much the same. And we also can divide bacteria in terms of their ability to, for example, make those amino acids, which have all these chemical requirements there. So they pick up this chemical environment from the food, from the atmosphere, like us, okay? Then again, they also require some of the trace element as we require. And we call them, for some of their enzymes, they have those trace requirements like zinc, magnesium, right? And other trace requirements they also require because they are cofactors of some of the enzymatic processes that are taking place in living things. Pretty much the same. Okay, now this will give you an idea that if you were to take bacteria and want to grow them, so many of bacteria, we term they use, for example, in this case, anaerobes or aerobes. So these are the terms that we use normally. Just give you a right idea that if you have a liquid material on the top is an air, you seal that tube and you want to grow the bacteria. Those bacteria, they want to grow in the air, right? They are going to come on top because they're going to take air over here. We call them aerobes. And since it's required for them for their food requirement to take oxygen from the air, we call them obligate aerobes. So they're going to love, they cannot grow without aerobes. And this is significant fact. If on the other hand, for example, they hate air. They can grow without air. They would rather grow at the basement, the bottom of the tube. We call anaerobes. They don't like oxygen. They want to grow without oxygen. And some of them can be mix and match. Some of them want to go over here, some want to go over here, right? Some of them will just need very minute quantities of either air or ice. So we have a classification based upon the capability of bacteria to either like oxygen or dislike oxygen. Okay. If I was to ask you, for example, a question, what do you think if you get infected with a bacteria that is caused lung pneumonia? What do you expect this bacteria to be in terms of their oxygen uses or consumption? Aerobic, because not anaerobic, aerobic, because they want to go and like oxygen and they want to live there, right? So that's what we say, aerobes and anaerobes. New version is available. Oh my goodness. No, I don't want that. Okay. Then again, I spent a little bit more time than I was planning to do. We need to talk about bacterial genetics because we want to understand how do bacteria pass the gene. And they were doing it for ages, right? Like human population. So basic, again, genetics, and I keep on saying to myself that I expect you to know some of the difference between DNA, RNA, and gene, and intron and extron. But keep in mind that most of the bacterial genome is single haploid chromosome. So that's very important for you to know. Then we talked the other day, they may have extra chromosomal genetic elements like plasmids. But today I'm going to tell you bacteriophages as well, and I'm going to explain that to you today. I also said the other cases that these specific extra chromosomal genetic materials are independent of the bacterial chromosome in most cases. So what I'm trying to say is, if one bacteria has a chromosome and divides into two, chromosome is going to split up into half. But if this chromosome has a plasmid, which is extra chromosomal DNA, that may not be transmitted. That's what I'm trying to say. Okay? Now, as I said, all the bacterial genome is, when you say genome, this means all the genes present over here, right? And they are present either in this chromosome. They may also be present in the extra chromosomal genetic material, but I said to you, by far, they may not be required. So they don't need to be distributed, okay? So there's selective advantage for that. Now, the genes are the sequence, pretty much the same definition. I don't want to go in detail, but you know the basic genetics, what is a gene, what is an operon, what is an intron. So the structural element of gene, how these genes are there, and what are genes made of. When you say you acquired 50% of your genes from your father and 50% from your mother. So this is you. So that's human, but we are male and female. So what happens in bacteria is, right, is only one. They split into 2 and 4 and so on and so forth. The chances are, we used to wonder that if that is true, where is the bacterial resistance coming from? But that information to deal antibiotic differently, if it's not in the chromosome, where is it coming from? So that was a question baffling microbiologists for ages. Then they found out there are plasmids there, extra chromosomal DNA. So then they figured out that's where it is coming from. That was the biggest discovery like a couple of years ago. Okay, now if you compare bacterial genes with us, we basically are diploid. So they are haploid, we are diploid, okay? And then again, bacteria have only one copy of their chromosome. That's the reason they are called haploid. And then again, if bacteria have only one chromosome, alteration of that particular gene, which is called mutation, will have a more obvious effect on the cell. So that's another common sense, that if you are picking up genetic material from only one source, like father, so if there's a mutation, that will be very obvious as compared to you're going to pick and choose for two. So then it's going to be a pick and choose, and you never know which one. You know, it's just like people saying, hey, your hair looks like your father, your eye looks like your mother. So there is a little bit of kind of choice that you may have. But in this case, bacteria would not have choice. They all have the same physical appearance for most part, okay? Now, the question is that this kind of the structural element, I remember telling you that is maintained by polyamines rather than by histones. So histones are what we need, those ball-like things I'm going to show you in a while, as compared to spermine and spermidine, which are polyamines. And why do we need to know that? Because we're going to target them, because we want to break it apart, because we want to kill bacteria. So that's the basis of antibiotics. Okay, recap. So you have a cell has a chromosome, and chromosome has this DNA. And these are histones. You see these histones? That is the one that will keep DNA in the format that this is, as compared to, this is human. In bacteria, they don't have histones, they have polyamines, spermine or spermidine. So they have a different structure as compared to us, we have a human. The question now comes, why do bacteria mutate? And again, if you remember when we talked about that, in cancer, I said there's a repair mechanism. We talked about RAG1 and RAG2 and many things. But what is the mechanism there? As a principle, there has to be an accurate replication of DNA, which is very important for survival of bacteria, because it has to transmit information in terms of what do we need to have for survival for thousands of years. So that's a basic part. We cannot expect, but again, it's a biological system where exceptions are already there. There could be mistakes, and accidental damage to the DNA might occur. Okay? But they have a very deficient DNA repair system as compared to human beings. So they don't have that kind of, repair system in terms of fixing the damage. So once there's an assembly line, whatever goes wrong kind of goes through. Remember in human beings, we had this apoptosis or so forth. We don't want to let those immature cells or immature things to go out. So, so the point is that if there's a mutation, change of the gene can occur. But good thing for bacteria is that most of these mutations have little effect on bacteria or are detrimental. And as a spin side is the other way around, which is bad for us. That kind of mutation that occurs may improve the chances of survival. And they can survive in a hostile environment. So we used to wonder if penicillin used to kill bacteria 10 years ago, why is it not killing today? Okay. The reason is mutation. Though it's a perfect system, but if there's a mutation, and that's what I'm going to spend time, how these mutations occur. Okay. The question again is that there's a gene exchange. You know, as I said, I give an example, human being, there's gene exchange half and half, father and mother. In bacteria, they're daughter cell. They should not have the gene exchange, the same gene, but we do see gene exchange. We call it prokaryotes. So what is happening is, and I'm going to use two terms today. Not all bacteria are bad, because we need bacteria for normal things, like I'd say, make bread for you, yeast, alcohol, fermentation, many good things. So there are good and bad bacteria. Not all bacteria are bad. And there are bacteria which are more dangerous, less dangerous. So we use the term pathogenic. So some of the bacteria that have a capability to cause disease, we call pathogenic as compared to non-pathogenic. Now, what we learned that this bacterial species, we call it promiscuous. So they are a bit promiscuous with their DNA, right? So if they want to invest their DNA or put their DNA some way, they're like promiscuous. They don't want to go for the regular channel, and we'll discuss in a while. So if that kind of exchange occurs between the cells, the new strains of bacteria will come. Okay, and this kind of promiscuous, we call promiscuous because it doesn't follow the norm. It is advantageous, and that's where the antibiotic resistance is coming. And it's not that simple. I'm going to go in detail, but I want you to understand and get this clear in your mind. Why is it that bacteria were killed by penicillin 10, 20 years ago, and why is it they don't get killed today? Because there's some information passed from to the progeny of the bacteria. Hey, this is what they're trying to give us to kill us. That's survival of the best. Let's defend that. And they pass this information. They pass this secret code to their offspring to survive. That's what they're surviving. Okay? And that's the code we need to break. That's our challenge in today's use of antibiotic is that they pass a code, we want to break the code. One of the things that we have to learn, as I said, is plasmids. Because we know for sure, I just told you, they're extra chromosomal. They need not to go into division bacteria. But then the question you can ask me, how they are transferred. They're not going to transfer for sure, right? If they are hanging around loose and you split it two, it may go in one and miss the other because they are not splitting. Chromosomes are splitting. They happen to be there in the cytoplasm, okay? Now, we discussed yesterday that these plasmids are like circular or double-stranded DNA molecules. They have a base pair. So this is a special thing in plasmid that carries that selective gene. Remember, I told you two plasmid, one was F, fertility gene, and one carrying the gene for resistance. You can imagine that we figured out and we researched and find out that that gene is coming from plasmids. Then the question will come, those bacteria that have plasmids have the ability to cause antibiotic resistance as compared to those who do not have that. Okay. So there are different types of plasmids. One of the plasmids bring antibiotic resistance. Some of them even brings bacteriocins. Some of them are responsible for toxin, and some of them are responsible for virulence determinants. Virulence means the ability of a particular bacteria to cause damage. Some of them will have more virulence as compared to others. Some will be more pathogenic as compared. As a rule, most of the bacteria that are pathogenic will be more virulent. So these are some of the genes codes that are there in the plasmid that may or may not. You raise your hand, yeah. Well, let me come to it a little later. But by now, pathogenicity is a ability of the bacteria to cause disease. Virulence is the ability to cause damage. I mean, there are a little bit overlap, but that's the best I can give you right now before going in detail. Okay, now plasmids. I mean, I used to wonder. I used to have to tell my students that bacteria have sex. I mean, we weren't taught that, so things have changed over time. We wouldn't have believed at that time that bacteria would have a sex, but that's true. So the conjugated plasmid is a special plasmid that is carried via mating between two bacteria. So in this case, one becomes a male, other becomes a female, and they have a sex. And we're going to discuss that in terms of what is it and how does that happen. There will be dissimilation plasmids, and there are factors I told you about the antibiotic resistance. I used to show one of the original paper of, I don't remember the scientist, when she published that. So she had kind of one paper, Valentine's Day or something like where, one bacteria is trying to kind of court another bacteria to bring that bacteria closer. So that's there. We call it pheromones. In human, it also happens. So there are some factors released by bacteria who will sexually attract other to come and mate physically. But anyway, so this is how the plasmids look like. You see this circular thing? And we can basically have a transcript of that. So we pretty much know very precisely what the structures are, what the genes are, and there's enormous amount of knowledge over there. I don't want to go in detail, but just give you a gist of it. Okay, now, those of you who haven't read the chapter so far, just start thinking of whatever I've just told you. So we know that we have a problem with antibiotic resistance. We know there's a plasmid there, and we are talking about division of bacteria. But the question is that how is it, and what are the possible ways that kind of a plasmid can be transferred? Okay. Now, what we found out that when bacteria get infected by viruses, so these viruses come and they inject their, and those of you who are interested, they can actually, if you Google how does bacteriophage infect a bacteria, you will exactly see it's sitting over there, presses his legs down, inserts the DNA or RNA there, and that kind of mixes up the bacterial RNA and DNA. Right? So that is there. And what we found out once they get infected, right? So bacteria, viruses, because remember, as a rule, viruses cannot survive on their own. They used to use their parasite, use protein for proteins assembly, and for their growth, they need to survive on another cell. So they survive on the bacteria, and they kind of divide over there, and then they split. When they split, by lysing that bacteria, so they will pick up some of the genetic elements from the bacteria. Okay, so they'll pick up a portion of those plasmids as well and pass it on to other bacteria because they're going to infect more. So we found out one way where a plasmid is transferred from one bacteria to other, not by binary fission, by bacteriophages when they infect and divide within the cell, and when the cell lyses, because I'll teach you in viral multiplication, cell can release bacteria or it can lyse the bacteria. So if, for example, lyse the bacteria, we call that kind of bacteriolytic infection, as compared to where they release without killing the host, we call lysogenic. So we know which bacteria are there. So some bacteria, while they are processing viral information, they will break apart, and some of them would not. But nevertheless, they will pick up the genetic material and infect another bacteria and pass that code to another bacteria. So we found out the code that goes from one bacteria to another bacteria, one of the mechanisms could be a bacteriophage, which is a virus. Okay. The other important thing we found out, we call transposons. So these are some of the things that I'm kind of putting in your mind. So transposons, where we call that jumping gene. And these jumping genes are present both in prokaryotes bacteria and us as well, where genetic information can jump. We call it mobile genetic element. They can transfer DNA within a cell from one position to another. So if you see the structure for that, so there's a gene over here and gene over here, so gene can jump around. So there's another mechanism we found out, the jumping gene we call transposons. So transposons can jump from here to here and then mutates. Remember, everything is coming from mutation. If it is a simple transposon, jumping gene, right? And it can just, because what it will do is just take up information from here and put it over there. Taking from here, so we call this gene running away with this information of gene from one place to another and insert it in another place. So we call it, like in this case, a simple transposon, which will just carry one gene and insert in the sequence, as compared to complex transposon, where it is going to carry different genes, steal different genes from one place of the bacteria and insert it into the other part. But when it does so, the bacteria sometimes dies if these transposon mess up the basic survival gene. For example, if there's a gene to make a good cell wall, and if that's messed up by transposon, the cell is going to die. So we can also use that. We can use a technology where we can make a bacteria a drug where we can introduce the transposons like a jumping gene. Because remember, when I told you pathophysiology, we have a gene gun. So we can, for example, if a person has a genetic disease, so that sequence of gene is missing, so you will grade that gene, put it in the gun, and give it to you so that it can be inserted. So we have that genetic technology in genetics today. So we are learning from bacteria that bacteria normally has those jumping genes. Okay, it's called transposons. Now, by the end of the day, the question that I'm going to ask you and the question I'm going to discuss is that there is a genetic information, a genetic code sent from one bacteria to another bacteria to become antibiotic resistant. And that is coming in genes. And we today, this far, we know there are three mechanisms for that. And this is very important for you. I remember Dr. Gentry was telling me, he asked this question to the class when he teaches antibiotic resistance. What are the mechanisms where the genetic material exchange between the bacteria? Right? And these are the three. Number one, conjugation. As I said, bacteria having sex. So that's a conjugation. The gene will be conjugated gene. And there is transformation. Bacteria can transform, right? And the third will be transduction. And I'm going to take these, remember these words, conjugation, transformation, and transduction. These are the three mechanisms that we have in bacteria. So conjugation, simple of all, which is a one-way transfer. So there is a donor cell we call a male to a recipient cell we call a female, and that happens to one of a pili we call sex pili, right? And that's one-way transfer of the gene. Okay, this type of conjugation happens between members of the same species or related species. That's important. So when it is a conjugation, physical contact between the bacteria, where one will become a male, other will become a female, or you call a donor to a recipient, and the transformation is called conjugation. And all genes is called conjugative genes, and the process is called conjugation. But the important thing is that typically occurs between the same species. So between, for example, say Staphylococcus aureus, so that kind of activity will happen, or related, which are common. Now, a little bit detail for that. As a rule, the donor bacterium has to carry a plasmid, which we call F plasmid, and F is for fertility, and it's male. So that carries 1+, we call that male because that's a donor. As compared to the other bacteria, we call female because that bacteria is a recipient, so we call F negative, and she is a female. So that's a conjugation between different bacteria. Now this plasmid, which is a conjugated plasmid, integrates and inserts through the physical contact of one of the pili, which is called F pili, to transfer that into the gene, right? So the F plasmid from one basically integrates into the chromosome of the other. So that's the important thing, is a chromosomal kind of a mix and match that normally happens. It's just like human beings, where you have sperm and you have egg, and then genetic material is kind of split half and half, okay? So same thing happens in bacteria. So when the bacteria, for example, there's a recipient bacteria which takes the positive plasmid from the donor male. Now this particular bacteria, we call it as a high frequency recombination state. It goes through a state we call high frequency recombination state, right? And this is a process which has to occur, because unless there is a sexual union between these bacteria, they cannot do so. So it has to go into the activation phase, where the donor will transfer whole pieces of chromosome into recipient bacteria. So that's the key over here. But the point I'm trying to make up over here is that in this case, plasmid from the male bacteria will be inserted into the chromosome of a female bacteria. Does that make sense? And I'm going to show it in the picture as well, so you'll have a better idea. Okay, now, if the plasmid sequence that we talked about, is excised from the host chromosome, it may take some. So remember, we have this ability to play with that. That's another important thing you have to keep in mind, that we have this ability to do that. So we have this technology to take this kind of, that particular sequence, because I showed you the plasmid sequence, right? So we can play with that. So we can take things out, and we are still working on that. But we know that conjugation carries that particular code for antibiotic resistance. We know that, and which is called conjugative R. I'm not going to teach you the detail, the genetic exact code, but just keep in mind that that's a process over here. Okay. Now, and I'm going to show, I think pictures are going to come later, but you may have to either look at the picture ahead of me or wait till I come to that level. So what is happening is that if you were to make this mating between two bacteria, one is male and other female, so we have the one we call male was the one that had fertility factor plasmid F+, as compared to the one which is negative. Because if the other is positive, that union cannot occur. So then in the end, what will happen is that they will form two F positive cell. By definition, so the donor gives F factor to the recipient. Now both of them become F positive. So in a non-technical way, it's just like both become male. Okay. Now, if the whole factor F is transferred from F positive to the recipient, that's normally happen. Or what may happen is that it kind of just keeps a copy for itself and pass it on. So there are two things that may happen. So this F bacteria can either give everything to the, or it can just copy it up, keep one, and pass it on to the other. So that's another thing that you have to keep in mind. And then again, when it comes to the F factor being inserted into the cell chromosome form HFR, most of the time what happens is that only a part of donor chromosome gets transferred, right? And this requires homologous recombination, And as I said earlier, it depends upon also the chromosome. Now, what I'm trying to say in this little bit complicated is when that union between plus and negative happens, the F positive can either copy that plasmid and insert it as a plasmid to the F negative one, or it can take the whole F positive and insert it into the chromosome of F negative. Right? So what will happen is, if that's what it does, that we call it high frequency state. That's a high frequency state in terms of bacteria. Let me see if, okay, there you go, finally. Okay. This is the only time I'm going to tell you the bacteria having sex with each other. But you have to remember that. Okay, so this happens over here is that there is a male bacteria, there's a female bacteria, F positive. These are the two chromosomes. Remember, they're extra chromosomal things. Keep that in mind. So it carries the F factor, and they have a pilus, we call it sex pilus, and that is inserted into F negative. And there's a tube that is formed, and that tube now inserts that over here. Okay? Now remember, as I said, one of the mechanisms that may happen that this one decides to make a copy of it, right? And keeps one for itself and passes the copy. So we'll end up with two F positive. So technically, in microbiological term, we'll have two males. Make sense? As compared to the other one, where the same conjugation may happen, where that plasmid is incorporated into chromosome of that, right? So it's gonna break it open and insert itself, right? And remember, they're all enzymes involved. I'm not teaching the enzyme. I'm gonna go detail molecular biology. But you can see if that happens, in this case, is gonna be inserted over here. So this cell, which has a F factor being inserted as a part of chromosome, we call HFR. That's one of my favorite questions. I can tell you right now, difference between an HFR and the other cell. Because other cell will just keep a copy as a plasmid as compared with HFR, which is going to incorporate that antibody resistance into the chromosome. Okay. Pretty much the same thing you can see from here. Conjugation, we already discussed that. The second thing is transformation. So in transformation, what is happening is that bacteria takes naked DNA. So the bacteria picks up a negative DNA from somewhere and tries to incorporate into its genome. So there is a DNA running around, bacteria picks it up, and some bacteria are very good at uptake of that DNA. And these are the most important bacteria: Haemophilus influenzae, Streptococcus pneumoniae, Bacillus species, and Neisseria. They are very capable of taking up exogenous DNA. So DNA is loose running in the atmosphere. So they will grab this DNA, put it into the sequence. So that's called transformation. So most important question that you need to know is that you know the names of these bacteria. Because if these bacteria develop antibiotic resistance, they are going to develop antibiotic resistance not through a process of conjugation, but a process of transformation, because they have the ability to pick up naked DNA from outside and put it in. And this is a good experiment that you can see which was done. The basic experiment, what they did was that they took a bacteria, for example, the one I told you earlier has capability to do that. This bacteria was injected in the mice, right? And you can see the mice died because you can grow the bacteria, and these are the same bacteria they were inserted. So what they did was that they thought for themselves, maybe there's a capsule. So what is that is killing the mice? Is it the capsule or is it the cell itself? So what they did was they took the same bacteria, unencapsulated, and give to these mice, and these mice survived. Okay? And then they, again, in the third experiment, the heat killed the unencapsulated mice and also found out that not only they died, but they were unable to grow as well. Then what they did was they put the capsulated and uncapsulated together to the same animal, right? And for their amazement, what they found was that only the one with the red one with capsulated were isolated. It may look very simple experiment, but if you already know, there was some mechanism that information was transferred from encapsulated to the uncapsulated one. So there was something going on where they were sharing the code. So two infections to the same mice, right? And one of them, because they were together, they passed this information, they transformed that. So what was happening was the killing bacteria over here took or passed this information to the non-killing one over here. And this also changed and transformed into killing because we could not grow the other species. So we found out that bacteria are passing this code from one another, transforming the other bacteria. So that was important experiment done at that time. Lastly, easy for you to understand is the gene transfer by transduction. I already explained to you. So virus goes and infects the bacteria and with all its genome. So it puts the genome that it brought from another bacteria plasmid, which carried the antibiotic resistant and put it into this bacteria. So this means you say, hey, this particular bacteria was not antibiotic resistant. Why all of a sudden this year it found out, oh, another bacteria become antibiotic resistant? Because that bacteria got infected by a virus bacteriophage, which carried that gene code and put it there. So that's the third mechanism we have in terms of transduction. And then there are all the different mechanisms over there. As I explained earlier, those of you who are interested, this is like a very good video. You can even ask me. But you can see from here, bacteria gets infected by a virus. So virus kind of puts this DNA over here. The DNA incorporates into bacteria. And when the bacteria divide to make new viruses, it's going to pick up that particular gene code and pass it on to these new viruses. So these new viruses now carry the code for bacterial resistance. And when they will go and infect another cell, they will pass it on to the newer cell. So this process of genetic transfer we call transduction. Okay? So I'll stop here, and I would